Hello and welcome. Some 500 years ago, we thought we understood the universe, and we did not. We knew a great deal about planetary positions, about calculating eclipses, but the Vatican astronomers particularly had the idea that the Earth was the center of the universe, and in that, they misunderstood a great deal. The point was that they knew a great deal, but didn't understand very much. It was thanks to people like this in the lower right, that is Nicholas Copernicus and later Galileo, to clarify things and give us a better understanding of what was going on in the universe and in science in general. Hence Einstein's remark here: any fool can know, but the point is to actually understand. We suffer from much the same problem with regard to aging. This is Dr. Leonard Hayflick, the first person to ever describe cell aging in 1961, and a friend of mine. And this is a remark he made when he and I were talking a year or so ago. And the point he's making here is that all of us, scientists, physicians, researchers, general public, would agree that the major risk factor for age-related disease is aging. But then we entirely ignore the aging process, and we focus on histology, signs, symptoms, biomarkers, outcomes, and downstream effects, rather than asking ourselves, how does aging really work? And the result is, we frankly don't get very far. So when we're looking at aging, we know a great deal, but we actually understand very little. We know a great deal about mitochondrial effects and effects upon any number of molecular cascades, but we don't understand how the aging process works, and that is a problem. So what I'd like to talk to you about today is age-related disease and the prospects for being able to cure and prevent age-related disease, which really depends on our ability to fundamentally understand the aging process. So the question I want to ask is, can we alter aging at its most fundamental level? And as you will see, the answer is not only yes, but we've been doing it for quite a while. We're just not as good as we'd like to be at it. This is a quote from a favorite poet of mine some 1,300 years ago, Li Bai, one of the very famous Chinese poets, who said, if you only hunt rabbits, tigers and dragons go uncaught. And indeed, that's exactly what we've been doing with regard to age-related diseases. We've been hunting rabbits. We hunt biomarkers, we hunt signs and symptoms, we hunt outcomes, without going upstream and asking ourselves, can we find the tigers and dragons and honestly deal with the aging process and do something about it? This is something that first came to my mind 50 years ago on this campus. This is Stanford, where I got my PhD and MD. And in those days, I started off looking at age-related disease and found myself frustrated because it wasn't clear how aging really worked. It took a number of years for me to get a clearer idea, culminating in some sense 25 years ago in me giving the first talk ever in history on the prospects for reversing the aging process at the fundamental cellular and genetic levels here at NIH in Bethesda, Maryland. And the point I made was that if we could truly understand the process of aging, how it fundamentally works, not just the biomarkers of it, but the fundamental process, we could cure and prevent age-related disease. And that resulted in me putting out the first book ever on the prospects for reversing human aging, fundamentally. The first articles ever in the medical literature in the Journal of the American Medical Association, and the first textbook ever, still the only textbook on this topic, with Oxford University Press, where I looked specifically at each and every cell system, different organ systems, and asked, how does the aging process work in these systems? For example, osteoarthritis and cardiovascular disease, dementias, and so on. And then a more recent book that the Wall Street Journal praised as one of the most important science books of the year, which is now out in a dozen different languages. In fact, just yesterday I was informed it's coming out in Turkish. But the point here is, can we do something about the aging process to make people's lives better? And that's really the issue. It's not a matter of increasing the lifespan, which we've done, at least with regard to the mean lifespan over the past 200 years, but in terms of making people's lives better. And while we have not affected the fundamental aging process at all in the past 100 years, we have increased the mean lifespan, if not the maximum lifespan, generally by a better understanding of things like microbial disease. And in fact, if we look back over those last 200 years, what we find is it's the advent of this conceptual revolution in understanding microbial disease that has allowed us to save lives. For example, the onset of hand washing in delivering babies drastically lowered both the maternal and neonatal fatality rates. Likewise, our ability to immunize and vaccinate, the first antibiotics, and so forth, have drastically improved the quality of human life over the past 100 years. Curiously enough, they have not done so by raising the cost of medical care. The general costs of medical care that have increased have been a result of incremental technical advances rather than fundamental advances like microbial theory. Example, in 1950, because of the cost of iron lungs, which are expensive and people were trying to improve them, and pulmonary care and nursing care in general, rehabilitation, braces, there were people who estimated that many industrial nations would be medically bankrupt by the year 2000. Yet by that year, the WHO found that the cost of preventing polio was a bit less than 10 cents per person. So much for medical bankruptcy. But the same sort of issue pertains now with regard to age-related diseases. There are estimates that, in fact, we will be medically bankrupt as a result of costs of treating dementias alone, Alzheimer's, for example, over the next 50 years. I think that the opposite is about to occur. As we truly understand the aging process in the same sense that we finally understood microbial disease over the past 200 years, as we understand the aging process, we'll be able to effectively intervene. And in fact, this has already been done, as you'll see a little later in this talk, with reversing aging in human cells, human tissues, and to some extent in mice. And I think what we're going to find is that as a result of this, not only do we improve lives as well as extend lifespan, but we actually lower the cost of medical care globally, nationally, and personally. Let me start by looking at a particular set of diseases, in this case, the dementias. These are ones that most of us are well aware of, particularly those of us who are clinicians. Alzheimer's dementia, Parkinson's dementia, frontotemporal dementia, and vascular dementias. What is interesting about these diseases is that while they are clinically quite real, there's a great deal of overlap between these different clinical syndromes. For example, Alzheimer's patients many times have motor symptoms. Parkinson's patients that generally is a motor problem often have cognitive changes. Likewise, frontotemporal dementias, and many of these patients, particularly when we look at autopsy, are shown to have vascular changes. So there's a great deal of overlap. And in fact, it's even more true when you look at sort of the alphabet soup of these. For example, Lewy body dementia, familial Alzheimer's, and medial temporal atrophy, and so on. When we look at these sort of dementias, you more and more realize that while they're clinical entities, they are also often atypical. There are atypical presentations, and there are mixed presentations. So over the past decade or so, a number of people have focused on this concept, which is there are age-related neurodegenerative diseases that are expressed in different ways in different patients. The same thing, as you'll see, is very much true of age-related cardiovascular disease and a number of other age-related diseases. So what is going on then is that we've begun to realize there are problems in understanding these age-related neurodegenerative diseases. And about 2 years ago, the Alzheimer's Association globally asked me to come give a talk in Washington for a conference I worked to help organize on animal models for the dementias, particularly Alzheimer's. Because the problem has been until now that, quote, everything works in mice, but nothing works in humans. And in fact, not everything works in mice, and the same problem pertains to dogs and cats and rhesus monkeys, baboons, and so forth. And what you find is the animal studies often have tantalizing results, but the outcome, as you see here, is sort of an empty bottle. And that's anakinumab notwithstanding more recently. So the question is, why is that? And I pointed out that there are technical reasons. For example, we don't share all the same genes that a mouse does. We have about 25% of genes, protein-expressing genes we don't share. And while we focus on technical abilities, for example, monoclonal antibody studies, and we have targets like beta amyloid, what we lack is an understanding of how these things actually work at the systems level. In fact, the problem is we're digging in the wrong place. And when you do that, it really doesn't matter how hard you dig, or how far you dig, or how much money you put into the NIH, for example, or National Health Service. The problem is you're not digging in the right place. We're focusing on outcomes, again, beta amyloid, tau tangles, mitochondrial dysfunction, and so on. And the problem is that those aren't fundamental to the disease. They are biomarkers, they're outcomes, they're signs and symptoms, and they're important ones, but they're not the process itself. So as a result of that, I was invited to write this paper that came out and generated more than 600 reprint requests in the first month or so. And why it caught people's attention was because what I was focusing on were systems issues and a unified model. And what I suggested that we needed was a unified model that could explain heterogeneity, for example, within the dementias. Why is it that you get Alzheimer's at age 50, and she gets it at age 80, and somebody gets it not at all? Likewise, even in Alzheimer's, you find differences in the presentations. For example, anamnestic versus patients who have less in the way of that, more cognitive function, problems with executive function. Also, the heterogeneity between dementias. Why is it that one person will get Parkinson's, another person will get Alzheimer's, the third person will get Lewy body? Why is it that Lewy body is different than Parkinson's? Why is there's heterogeneity? Also heterogeneity between species. We know that any species we look at shows cognitive decline with age, but they do so in different ways, both clinically and histologically. If we look at mice, for example, the way they handle beta amyloid is not the same way that a human Alzheimer's patient does, and yet there are cognitive declines seen in all these species. So can we explain that? We also need to explain upstream mechanisms. For example, we know that having two ApoE4 genes increases your risk of Alzheimer's, but why? We know that exposure to some toxins, for example, paraquat or dioxin, increases your risk for a number of dementias, but why? We know that head trauma does, for example, closed head injury, traumatic brain injuries, increase your risk of certain dementias, but why? Now, likewise, we know that downstream biomarkers, we know a great deal about many of them, dozens of them, maybe hundreds, but certainly beta amyloid, tau tangles, mitochondrial dysfunction, and so forth. But why do these occur? And why do they change in different patients at different times? So we need a unified model that can explain this. And this is the kind of model I was offering. Now, I was also saying that perhaps more importantly, we need a systems model. And let me explain what I mean by that. What I mean is we do not need to focus on components. We need to focus on the system itself, the dynamic system that results in these diseases. You're looking at the cockpit of an Airbus 380, and in the upper right, you're seeing a Pearl 15 Rolls-Royce jet engine. In the lower right, you're looking at a Trent 900 turbofan that powers the Rolls-Royce jet engine that powers the Airbus. And you're seeing a small fan blade in sort of a turquoise blue. And when that fan blade fails, the appropriate engineering question is not what's wrong with that particular fan blade, but what's wrong with the way that fan blade functions in that turbofan, in that Rolls-Royce jet engine, in that Airbus 380 at 38,000 feet, going hundreds of miles an hour at a high RPM and a high temperature. In short, it's a systems question. It's not a component question. It's not a question of that particular biomarker. It's a question of how it works in terms of the whole dynamic, complicated system. Same is true in medicine. These are typical signs and symptoms of COVID infection, but they're not COVID. They're biomarkers, they're outcomes, they're signs and symptoms, they're downstream findings, but they're not the disease itself. If we truly want to understand and cure and prevent a COVID infection, what we need to do is not delve into the individual signs and symptoms listed here, but we need to understand that it is a complicated interaction between a virus and the human immune system and the rest of the body. And it's not until we understand that we can do something about the disease. The same is true of aging. At a macroscopic level, these are typical diagnosis or biomarkers, many of them, for age-related changes that we see in ourselves and those around us. But they are not aging. I don't care how much you know about gray hair particularly, you need to understand it in a more systematic and fundamental sense of how this works dynamically. And the same is true of all of these other diseases. To understand, for example, cholesterol plaques in arterial disease is not sufficient. We need to go further. The same is true when we look at the microscopic level. None of these things are aging by themselves. Telomere shortening, acetylation changes, lipofuscin, declining rates of ATP to react to oxygen species, DNA repair changes. None of these are aging. They are biomarkers of aging, but they're not the process itself. They're not the dynamic system, and that's where we need to go. Same is true if we look at some of the dementias. These are typical findings in many dementias. For example, beta amyloid in, for example, Alzheimer's disease, tau tangles, and many other dementias. Changes with alpha-synuclein seen in, for example, Lewy body and Parkinson's, blood-brain barrier failure, lymphatic changes, but none of these are a dementia. They're biomarkers, they help us define it clinically, but they do not define the process itself, and we need to go further down. Same is true when we look at these things genetically. Knowing, for example, that if you have two epsilon-4 alleles, the ApoE4 alleles, increases your risk, doesn't tell you how they work, doesn't tell you about the system. The same is true of presenilin genes and the more than 200 other genes I could list on here. So it's not enough to know the genetics, which do not show 100% penetrance nor 100% protection. We need to understand how they work in a dynamic context. We need to understand those in order to be able to treat and prevent Alzheimer's and other dementias. Bluntly put, unless we understand the aging process itself, then how can we understand age-related diseases? And more practically, from a clinical perspective, how can we actually cure and prevent such diseases? So let's look at aging for a minute more closely. These are species most of you know about. Human beings in the upper right tend to live about 3/4 of a century. That is Coco in the upper left, the gorilla that I had close contact with for about a year because I was her babysitter. She had a bigger vocabulary in sign language than I did, but a lot of fun to play with. She lasted a little less than a half a century, which is typical for the gorilla. Typical domestic animals last a little less than a quarter of a century, again, depending on the breed involved. Typical rodents last 2 or 3 years, again, depending on the individual breed involved. Now, the question you might ask is, why do these creatures age at different rates? Clearly, it is not a matter of entropy. It is entropy in the context of genetic differences. But saying that does not tell you how it works. We know, for example, that, as I say, mice share about 75% of protein expressing genes with us. So something is peculiar about that other 25% of protein expressing genes, to say nothing of the regulatory components. But the question is, what? It's not enough to say we vary genetically, and that's why we vary in terms of lifespan, without understanding something deeper. The same is true if we look within a species. For example, these are Hutchinson progeric children, and I gathered this group together, and I used to every year gather these children together all over the world. In this case, we have taken them for a tour of the White House, which was quite a lot of fun until the Secret Service asked us to leave because Sammy stepped on the pressure sensor inside on the stairs up to the presidential quarters. But these children die overwhelmingly of age-related cardiovascular disease. The typical lifespan is about 12.7 years. And the question is, why do they age so much faster than most of us do? Now, we know the gene. It appears to be the lamin A gene is abnormal, but that is not an explanation, and it certainly doesn't explain aging. What it does is tells you where to look and where to begin, but it's not an explanation of aging any more than biomarkers are. The same is true, curiously enough, when we look retrospectively. For example, every cell in your body comes from a germ cell line that's been extant on this planet for something like 4.5 billion years, depending on who's estimating. And every mitochondria in your body and eukaryotic line goes back about 1.7 billion years, again, depending on who's making the estimate. What's curious is that retrospectively, that cell line has not shown any evidence of aging over time. And yet clearly, the cells and mitochondria in your body show age-related changes over the past few decades. Now, it also depends sort of where and when you look. It turns out that after fertilization, there are some changes that you can look at in the germ cell line of mitochondria that show some aging and then some correction. And what's really going on is an active process in which entropy occurs, but there is effective maintenance. In fact, the same is true generically. If I look at the germ cell line, what you find is that maintenance is essentially 100%. It's dynamic, changes over time, but essentially it keeps up with entropic changes. The same is not true of the cells in the 90-year-old man's hand there on the left, the fibroblasts, keratinocytes, melanocytes, hair follicle cells, and so forth. Those cells do not have as effective a maintenance as the germ cell line does. And the end result is age-related changes, in this case, in the skin. In effect, what you're looking at is the ability to keep up with entropy. And as Alice in Wonderland said, or the Red Queen said in Alice in Wonderland, we must run as fast as we can just to stay in one place. That's what's going on in cells that maintain themselves over time, for example, the germ cell line, and in subsequently in regard to mitochondrial function, as we'll see. So aging is not a matter of entropy. It's a matter of the failure of maintenance in the face of entropy. It is not entropy alone. And it's a common mistake people tend to make, and it prevents them from getting very far in terms of age-related diseases. We can look more carefully down at lower levels. And what you find is that for any molecular pool in your body, whether it's, for example, collagen, elastin, beta amyloid in my brain, tau proteins, myoglobin, wherever I look, you find that there is no molecular pool that's static. It is dynamically maintained. It's continually being turned over or recycled, if you will. Now, in effect, it's running as fast as it can to stay in one place. Now, the result of that is you are continually making molecules, degrading them, making them, degrading them. And when there's damage, signified here with a little red star, it tends to get washed out in young cells. But as we get older, the rate of molecular turnover slows. And the result of that is you begin to get a little accrual of more and more damage because you're not keeping up with it. You're not, in effect, running as fast as you can to stay in one place. And in older cells, it's particularly true. So the percentage of dysfunctional molecules goes up no matter where you look. And this includes lipid membranes, includes everything you can think of. You can even look at this mathematically, for example, from my Oxford Press textbook. And really what it's saying here is that young cells with a rapid rate of turnover have very little damage accrued at equilibrium, whereas older cells with a slower rate of turnover have a larger accrual of damage at equilibrium over time. And the result of that, as we'll see, can have effects on the way things like the brain work. We could look at other organs, but we're going to take a typical one for me, in this case, the human brain. Now, where we stand right now is that everybody has been chasing rabbits. They're going after monoclonal antibodies. Anakinumab just got released a few weeks ago. There are a number of monoclonal antibodies here in red that have been declared not working, a number that are still in the pipeline, and a couple that have gotten expedited status. But again, they're not looking at things from a more dynamic perspective. But let's look at what their major focus is. Usually it's beta amyloid. Even if we look at beta amyloid, in this case, the way it's dealt with by a microglial cell in the brain, you find you are continually producing beta amyloid, secreting it, rebinding it, re-internalizing it, degrading it, producing it, secreting it, and so forth. It's a continual recycling process in young cells. But again, as the cells get older, what you find is there's more and more chance for these molecules, for example, to cross-link and degrade externally. And the result is beta amyloid microaggregates. Now, you might reasonably ask, what about the genetic predilections? For example, if I have two ApoE2 genes, two ApoE2 alleles, I tend to make molecules that aren't very sticky. So even as my turnover of beta amyloid slows with age, I don't get much in the way of microaggregates because they're not sticking together. But on the other hand, if I have two ApoE4 alleles, what you find is I've got at least double the risk if I have one allele. If I have two alleles, 3 times the risk, and we tend to get an earlier onset of Alzheimer's. And the reason is people with ApoE4 alleles tend to make stickier molecules, so you form microaggregates more readily, even at a younger age. And the outcome for all people as we get old enough is you begin to get beta amyloid plaques, depending on other risk factors you've got, genetic, environmental, and otherwise. But it's the slowing of the beta amyloid turnover. And the same is true whether we're looking at beta amyloid, tau proteins, mitochondrial changes, as we'll see, inflammatory changes, and so on. All of these things have to do with cell senescence, changes that are going on as we get older in the face of genetic changes or genetic predilections as well. But the outcome in the brain is synaptic loss, dysfunctional neurons, necrotic areas, and frank clinical dementia. Nor is it simply a matter of those particular proteins involved, for example, beta amyloid and other proteins or the molecules. We also find that as a result of changes within the nucleus, changes in expression of a number of important aerobic enzymes that go to the mitochondria. Again, most 99% of the aerobic enzymes in mitochondria derive from the nucleus, and they slow down in their turnover rate too. And the outcome of that then is more and more dysfunctional aerobic enzymes. So your ratio of ATP to reactive oxygen species begins to go down. You make more free radicals and less in the way of power. Also, the membrane itself is being turned over less rapidly, the lipid membrane molecules. So the membrane is more porous. Also, the scavenger molecules, for example, superoxide dismutase, catalase, these are molecules that, again, are being turned over more slowly. So as they get, in effect, eaten up, they're not replaced as quickly, so they're not as efficient. And the end result then is more damage, and the recycling rate of those damaged molecules goes down. So for at least 4 separate reasons, which are multiplicative, you'll find that mitochondrial dysfunction increases with age, as well as reactive oxygen species damage. But it all goes back to changes in the nucleus in terms of the expression of important proteins. The same is true of DNA repair. The only molecule in your body you actually repair is the DNA, chromosome, the bases in the chromosome. And there are 4 major molecular families, you might say, for DNA repair to prevent mutation. That is, molecules responsible for detection, those for excision, those for replacing the base, and those for ligating it into place. And the turnover rate, the recycling rate of these families of molecules for DNA repair also go down with age. So you get an exponential increase in the rate of mutation, and that's clinical cancer. By the way, that's over a lifespan, not over years. So there is an exponential increase in the rate of mutation for almost any species over its lifespan, whether it's 2 years or in the human case, perhaps 75 or 80 years. It's not the years, it's the rate at which DNA repair slows. And in fact, what you find is that this has to do with telomeres, but again, telomeres don't cause aging. It's just that they are an indicator of what's going on with patterns of gene expression. And telomeres no more cause aging than genes cause evolution. They're a key player, but they're not a cause, which is sort of a poor choice of verbs here. Also, the length is irrelevant. It's not the length, it's the rate of telomere loss that makes a difference in the pattern of gene expression that changes with age. So you can actually plot, as has been shown here from an article that was put out by Maria Velasquez's group, you can plot lifespans based on the rate of telomere loss, because what's actually going on is telomere shorten as they change the pattern of gene expression, which changes the turnover rate of molecules, which changes the cellular function. In fact, you can look at it this way. What's going on is cell senescence. In upstream, there are a number of things that affect cell senescence: age itself, genetic risks, some of which I've alluded to, exposure to toxins, for example, again, dioxin, chemotherapy toxins, a number of them, trauma of any kind, whether it's trauma to your knees or in the case of dementias, trauma to your head. Hypertension and hyperglycemia are typical examples for vascular aging, and that includes vascular dementias. Likewise, other infectious agents, which can have an effect. And what these tend to do is accelerate the rate of cell loss, cell division, therefore telomere shortening, therefore the pattern of gene expression that the telomeres modulate. And the result of that is slower molecular turnover. And as a result of that, as we've seen, is you begin to get cell dysfunction, failure, and cell death. Now, the downstream outcomes are, again, lesion. I've listed a couple of dozen here, but when I'm focusing, for example, on dementias, I tend to look at the beta amyloid, tau tangles, alpha-synuclein, and so on. But all of these other listed here, DNA, the mitochondrial dysfunction, slower DNA repair, I've alluded to. And over the past several hundred years, we have tried medically to intervene in some of these cell senescent processes that result in clinical disease, but not very effectively. For example, we do know that if I normalize your hypertension and I make you euglycemic, I can lower your risk of cardiovascular disease or indeed of age-related vascular dementias, but I can't prevent it. I may slow it, I may delay it, I may make it a little better, ameliorate it, but I can't prevent it because aging still occurs. The same has been true, by the way, of downstream processes. Hence the issues I just talked about with regard to monoclonal antibodies and beta amyloid. None of these have been shown to actually alter the vector, the slope of the downhill course. You delay it slightly, the course after that becomes parallel, let alone can they stop it or reverse it. So we need something much more effective than just these sorts of approaches. And that brings us to the question of where's the optimal point of intervention to prevent and cure age-related diseases. And the optimal point of intervention is to do something about the pattern of gene expression, which is modulated by telomere changes. And the question then is, can we change shorter telomeres into longer telomeres, reset the pattern of gene expression? Well, as it turns out, the answer is yes. And in fact, it was already done. It's been first done 23 years ago by Andrea Bodnar's group out of Jaren in this classic article in Science magazine. And what they showed was that you can take old cells that have a pattern of gene expression and young cells which have a pattern of gene expression, very typical, and you can convert young cells to old cells simply by re-extending telomeres. The pattern of gene expression, the epigenetic pattern, reverts that to that of a young cell. So we have known for 23 years that we can reverse aging in human cells in the laboratory. So it's startling to most people, but that's the way the data is. The question is, can we do it with tissues? And the answer, again, is yes. This is Walter Funk's article. He was the lead on this, again, from the Jaren group, this one 21 years ago. And what they did was they showed that if you take young human skin cells, you can grow young human skin. If you take old human skin cells, you can grow young human skin, in this case, explanted onto a mouse, an immunocompromised mouse. But if you take old human skin cells and reset telomere lengths, you can grow young human skin on that mouse. So we know that we can take old human skin cells and regrow young human tissue. That's been shown, as I say, some 20 years ago. Furthermore, it works equally well when you're looking at bone cells. It also works when you're looking at vascular endothelial cells. Literally, you can take vascular endothelial tissue from an older person and regrow young arterial tissue. So the question is, can we do that with organisms? And the answer is yes, actually. At least to the degree technically we can do it at all. It was first shown now some 10 years ago by Rhonda Pinno's group at Harvard, but they used a technique in which they were altering the germ cell line. Now, this is another article out from Maria Blasco 9 years ago. She's the director of CNIO in Madrid. And what she did was use a technique that we can use in human beings using a viral vector to deliver a TERT gene. She showed that not only do you get a lifespan increase in these mice, but the physiology improves. For example, the bone mineral density, the subcutaneous fat measures of immunofunction, but also the behavior improves. These animals that have demonstrated cognitive decline go back to showing essentially young cognitive function. Which brings us to the question of, can we do this to human beings? So what I'm suggesting that we're about to do is not hunting rabbits, but hunting dragons. When it comes to age-related disease, our initial target is going to be Alzheimer's disease. I'd like to tell you a little bit about what we're up to. This is our scientific advisory board. They're going to push for doing this. These are, as I say, global experts in gene therapy, statistics, telomerase, and trial design. For example, Suzanne Hendricks there designed the Biogen study for the anakinumab. Kurt was responsible for some of that early mouse data from Maria Blasco's lab. Joe Araujo runs Intervivo. These are people with practical businesses and institutes who run these things. Joe is responsible for our being able to do dog studies, for example, with regard to dementias, knows more about it than anyone in the world. Our clinical advisory board, likewise, are experts in clinical trials, neurology, and Alzheimer's, have been running FDA human trials with Alzheimer's for quite a number of years. I want to emphasize that these people were not chosen because they have drunk the Kool-Aid. They were chosen because they're interested in rigor and credibility as we take this to human trial. They see the value in the model and want to see it tested rigorously, as do we. What we're going to do is we're going to use telomerase therapy. Our initial target will be the human brain. Initially, we'll be looking at Alzheimer's, but then we'll progress on to other dementias. We'll then go after the vascular system. But this is a platform therapy that can be used for the central nervous system, chondrocytes, osteocytes, renal podocytes, and so on. And we'll be progressing on to other tissues as well. Our priorities are not only safety and efficacy, but credibility. We're being very careful what we say and what we do with this because we want to make sure that the data stands on its own. The technique will be a single dose using an AAV vector that'll be custom made in a custom cassette and human telomerase gene. And that brings us back to the initial question I asked. Can we alter aging at its most fundamental level? The answer is yes. The question is, can we do this to human beings and make lives better? And I think the answer is yes also. Thank you very much for joining me today.